Hello, good afternoon, everyone. I request everyone to take their seats. We are right on time, 2:30. All right. Good afternoon. Thank you all for coming to this session. John Luke, who is co-chair with me, he's an associate professor at University of Rochester. And me, I'm Niraj from Johns Hopkins University, Department of Genetic Medicine. So why we are here for this particular session, as you can see from the title, this is Cystic Fibrosis Therapeutics when there is no protein made. What does that mean? Of course, first I have to give you the disclosure slides that neither John nor I have anything to disclose related to this presentation. So this is the problem that we have to address. There are many disease-causing variants that are identified in CFTR gene. Technology each day is improving, and we are going to get more and more of the variants known with the advent of whole genome sequencing that is going to be available at a very cheap cost. So what does that mean to the field of CF? What this pie chart here is showing is that even today, the majority of the variants are RNA processing, and why this is important. In blue here is shown a missense variant that constitutes 38% of the total CFTR variants. However, the major chunk of the remaining is RNA processing. We all are enjoying, and we have provided a hope to people with cystic fibrosis who harbor missense variants. Their lives have been transformed because of the drugs that are available to them today. But the job is not done. It is not done for these people that harbor such kind of variants. And what I'm again trying to emphasize on the point is that many more variants will come in these flavors. You will call them as nonsense variants. You will call them as splice-site variants, structure variants, enhancer variants, and the variants far from CFTR gene that we still do not know how they're going to regulate the transcription of that particular gene. We are not still talking about epigenetics and chromatin modeling. So with that, what we have got here, we have assembled 4 great speakers who are expert in their fields, and each one of them are going to touch upon the aspects that I have told you, and you will see in the years to come that this therapeutics will come and give some hope to people who are left behind today. With that, I'll ask my co-chair, Dr. John Luke, to introduce our speakers.
All right, welcome everybody. So just to give the format of how it's going to go today, the speakers will have 20 minutes to share what they have, their research. There'll be 5 minutes of questions, including some online questions. And at the end, we're going to ask all the speakers to come up for a short panel. So if you have questions for the first speaker that you thought of during the fourth speaker's talk, there'll be some time to ask those questions. All right, so we'll get going. So our first speaker is Margarida Amaral from University of Lisboa in Portugal. And the title of her talk is Timing is Everything, Kinetic Studies of Nonsense-Mediated mRNA Decay. I think
the title is still... Yes, I think it's like here. Right.
The title is...
So I must provide you with an explanation because I'm filling in someone whose title was what John said, and this is my title. So I must thank Niraj and John for actually putting so much confidence in me that they invited me yesterday to do this talk. So I'm going to talk about reading through the lines, smart molecules to rescue PTC mutations. and I have no presented disclosures on this topic. So today we've heard a lot about precision medicine, what I like to call theranostics, because is diagnostics for the best therapy. And that's exactly a big challenge, because we need to treat individuals more precisely and effectively in ways that meet their individual needs. So if we have a group of patients, in the classical discovery for drugs, we carry out, or pharmaceutical companies carry out high throughput screening, and then they come up with one drug, which is like one drug fits all. Of course, the largest group of individuals will get benefit, otherwise the drug is not approved, but there will be some individuals who don't get benefit and some who actually get adverse effects. So in the new concept of precision medicine or theranostics, we should have some sort of biomarker which allows us to pre-triage individuals according to the existing drugs so that each patient benefits from an individual drug. So these are the functional classes of mutations which have been called or evolved into theratypes. You must notice that we have here a class 7 mutation, and the reason is exactly because these classes of mutations have evolved into theratypes. So the respective strategies to rescue each class of mutations have been highlighted here. And because this class of mutations is very different from this one here, which includes the so-called PTC or premature termination codon, so the mutations with the small x, like G542X, R1162X, and so on. So it is believed that they can be rescued by using read-through molecules, also termed suppressors. Whereas these ones, like large gene deletions, some of them that Niraj actually mentioned, like splice-site variants, which cause frameshifts and so on, can only be rescued by targeting other channels that we have, or gene therapy or mRNA therapy. Okay, so the focus of my talk today is actually on these bona fide class 1 mutations, which are the PTC mutations. So what do we have so far on those protein rescuing drugs which are approved? So most of the individuals who have at least one copy of F508del, they can benefit from 2 correctors and 1 potentiator, the so-called triple combination drugs, now already 2 in the clinic. Individuals with gating or residual function mutations, they can benefit from just 1 drug, ivacaftor, and the other individuals who have neither still don't have a drug which actually modifies the phenotype at its root to benefit from. And the 20% I put here actually are data from the European registry. I believe the number is lower in the US. So of these mutations, at least 80% have these premature termination codons. And what do these variants or these mutations actually cause? As the name indicates, the premature termination codon acts like a stop signal which directs the synthesis of the protein from the messenger RNA at these small factories, which we call the ribosomes, and everything is released. And because the cell has surveillance mechanisms, namely the nonsense-mediated decay, or for short NMD for messenger degradation, these messengers bearing PTC mutations actually are degraded. And of course, if they are not fully degraded, the remainder amount originates truncated, that is, shorter proteins. So the aims here are twofold. Avoid messenger RNA degradation by finding molecules that inhibit NMD and suppress premature stop codons with suppressor, that is read-through agents that sort of mask this premature termination codon. So very briefly, our current model for the decay steps of RNA-bearing PTC mutations is focused mainly on two steps for substrate recognition, meaning RNAs which have PTCs. One is the presence of the EJC, or exon junction complex, when it's downstream the stop codon, or to the 3' side of the messenger RNA. And in sometimes also a structure, so a big structure of this region of the messenger RNA, which we call the 3' UTR. Okay, so in the past, in our lab, we have found out the relative abundance of messenger RNAs bearing PTC variants versus this is not exactly a missense, but it's F508del is actually a deletion, but versus the levels of F508del. And if you can find if you can read the numbers here, you will see that almost all are drastically reduced, because these are the percentage of remainder as messenger RNA. With one exception, which is when the stop of the PTC mutation is at the very beginning of the gene, which avoids somehow this process of NMD, but that's altogether another story. So all PTCs except if at the start of the first part of the protein undergo NMD, nonsense-mediated decay. And we also have looked the levels of other variants, missense variants, versus F508del, and there is virtually no change. So it's almost 50-50. So they do not undergo NMD. The previous data were on nasal cells, so respiratory cells, and here are data from intestinal organoids. And we can see pretty much the same, and the messenger RNA, which is still in higher levels, is again at the first part of the gene. So all PTCs, mRNAs undergo NMD, both in the airways and intestinal epithelium, but if there is no PTC, they do not undergo NMD. So what sort of drugs are we looking for? So in this premature termination codons, or stop codons, which lead to NMD, as I said, and to truncated protein, we aim to find drugs, and these can also be suppressor tRNAs, as we have already heard in previous sessions today, that actually avoid the degradation of NMD, so inhibitors of NMD, and read-through agents to lead to the production of full-length protein. In the past, there were a few drugs actually trying to achieve this goal, PTC124 or Ataluren. Also, the antibiotic tobramycin has some of this effect, and ELOX 02, but they were discontinued for CF. So what we have put in place is an assay for high-throughput screening. It's actually sort of complicated construct with 3 tags, mCherry, so red label at the which will result at the N terminus of the protein, EGFP at the C terminus of the protein, so 3' of the RNA. Some introns, so it's a mini gene because, as I mentioned, the process of NMD is tightly coupled to the splicing complex, and also a flag tag. So what are we going to expect from here? So 4 possible results. No effect, so we have very basal fluorescence. Enhanced mCherry if there is inhibition of NMD, so high red, low green. Enhanced mCherry plus enhanced GFP, so there is read-through as well as inhibition of NMD. And if the amino acid which is included instead of the PTC actually enables folding and traffic of the protein to the cell surface, we can detect it by usage of a flag tag in the protein detected without cell permeabilization. So in collaboration with chemists from our department, actually, they have been synthesizing molecules under two chemical scaffolds and different groups, so variations of these compounds. And we have now analyzed series 2 and 3, also 4, but I'm not going to talk about 4. There's nothing much interesting occurring there. So these are the results of the microscopy high throughput screen, and these are the labels. Of course, you cannot read any of this. It's just to show that we have performed the screen. And the cells were treated with these compounds at 3 doses, 2, 5, and 10 micromolar, and imaged 24, 48, or 72 hours after incubation. So we measured also the autofluorescence of the compounds, because this could impair the readout of the cells. So I have here 2 examples of compounds. Here is compound 19, and you can see that there is significant increased fluorescence, not here, this is just the nuclei, but in the mCherry and GFP for this compound 19. For compound 17, we also see some GFP and mCherry, and a little bit increase, not significant, for the flag tags, so the protein at the plasma membrane. So compound 11 only shows PTC read-through. It's none of the 2 I'm showing here. Compounds 12, 17, and some in the 4th series have both PTC read-through and NMD inhibitor activities. Compound 19 only NMD inhibitor activity. Okay, this is just to show toxicity, and out of the 7 best compounds, 6 had actually no toxicity in the 2 to 10 micromolar range. So going quickly, compound 11 in the 2nd series actually was the one showing higher levels of toxicity at 5, 10, and 20 micrograms. So now we check the effect on different variants, so in the first part of the gene, in the middle, and in the end for these 3 CFTR variants. And we analyzed the data by qPCR, quantitative PCR, in both the 3' end and the 5' end, and of course when the levels of messenger RNA vary, as you can see here, there is significant NMD in these 2, not so much in the Y122X. So the 3 of them induce NMD, which we already know. So by testing the effect of the series 4 compounds, I told you they're not interesting, I'm not going to talk about them, but the most effective and pronounced effects were actually on the blue variant, which is Y122X, and for compounds in the series 2, 11, and series 3, 19, as shown here. But actually on the other variants, also some effect was observed. Then we wanted to see the additive effects in combination with what we call the controls. So SMG1, which inhibits NMD, and G418, which is a read-through agent. And we have here the 4 of the top compounds from the screen. So CMG1, as I said, is an inhibitor of NMD. G418 is a read-through agent tested in the laboratory. So we observed statistically significant additive effects for compound 11, which we had only observed with read-through activity, and together with NMD inhibitor SMG1. And compound 11, for which we have found both activities, it's actually interesting because its effects are additive with both SMG1 and G418. So very preliminary data on Western blot. and here for the W1282X variant, what we observe for compound 11 at both 2 and especially at 5 micromolar is a significant stabilization of, we believe it's band B with the appearance of a small amount of band C. So because checking on time. Because we want to move this closer to physiological models, I'm going to introduce the models that we use. So usually cell lines are used for what we call theratyping, and then patient-derived models like, for instance, induced pluripotent stem cells or primary nasal or bronchial respiratory cells cultivated in the lab, or organoids are actually what we can call patient-specific theranostic models. So here we focused on these two types of models, actually in collaboration with Jeff Bickman for the intestinal organoids in Utrecht in the Netherlands, and with Nicoletta Pedemonte in Italy for the primary cells. So what Sasha and Lara in Jeff Bickman's lab found for these two compounds, 11 and 12, in intestinal organoids, okay, they are very robust because we're also trying to work on nasal organoids and to the significance in nasal organoids. But in intestine, they actually tolerate quite well. So the toxicity was not observed here for compound 11 and also compound 12, which we did not find toxicity. So on their own, there was not significant rescue of each of these compounds, neither at low dose with together when they were together, but at higher dose there was a significant increase in the swelling, meaning that they were able together to rescue this variant. This is, by the way, homozygous for this variant. Together with ETI, it probably became too toxic because the effect disappeared. So with Nicoletta data, and these again are preliminary data for a different variant, R553X in heterozygosity with F508del, we have here that the compounds individually do not do significant effect, but each of them with ETI, together with ETI, are actually able to rescue this variant. So this is one donor and preliminary data, but they found that both compounds are additive to ETI. So to summarize what I've shown you today, for class 1 mutations, the bona fide class 1, so excluding the class 7, these are the ones with PTCs, is that we observe that all messenger RNAs undergo nonsense-mediated decay, except if the mutation is at the first part of the gene. Of course, non-PTC mRNAs do not undergo NMD, and the rescue drugs that we look for are both PTC suppressors and or NMD inhibitors. So the best novel compounds so far are compound 11, which apparently is only an inhibitor of PTC, but exhibits in some cells some toxicity, and compound 12, which seems to do both effects. In intestinal organoids, we observed some synergism with these two compounds, so leading to think that they have some distinct mechanism of action. And the primary cells tests, although preliminary, and they will help to prioritize the development of the drugs, have shown that the two compounds are additive with ETI, so each of them separately with ETI. And they also observe the same with ELOX. But there is inter-individual variability and possibly also between variants, CFTR variants. So these are the PIs in this UK CF Trust grant that we are working on. Mike Ray is the coordinator in Newcastle. Cristina Moiteiro is the chemist in our department. Nicoletta Pedemonte in Genoa, Italy. Jeff Bickman in Utrecht. And we have other collaborators and, of course, funding. And these are the groups, the 4 groups working on this SRC. And I thank you very much for your attention.
On time. That was gold standard.
That was great. A note to all the other speakers, that was perfect timing, so if we can continue that, that would be great. If there's any questions, please come up to the mic. Go ahead on the left here.
This great work. So I just wonder, so you talk about the intensity organoids, maybe it's different, the mechanisms. Do you think is test system reason for organoid system or the different cell, the efficiency is different, is cell system reason or is readthrough mechanism? Could you talk a little bit this, you think about this difference mechanism, the readthrough?
It's hard to hear because of the reverb.
Yeah.
If you can come up, because the reverb's really difficult.
Yeah, there's echo in the room.
If you can.
Yeah, I just wonder, you talk about the readthrough efficiency in the intensity organoids is very high. So that's why you talk about is maybe is a difference readthrough mechanism.
Maybe you could
Just come up and talk to us. We'll get to the second question. Come on up. Walk up to here.
come here.
Come here.
We don't bite.
This,
Yeah. Then the second question, So the difference in cell types, is it because of the different cell types or toxicity or function?
Or mechanism. Yeah, maybe I repeat. So is it the differences that we observe in the different cell types, organoids, and respiratory cells a consequence of different cell types or different mechanisms of the drugs? That's a very good question, but at this point I cannot answer, and it's probable that, the results are still sort of preliminary. And in my opinion, there could also be an additional variability, which is the context of the PTC variant. So not just the real stop codon, but the sequence context of what we're trying to read through.
Dr. Sosne.
Thanks, Margarita. Patrick Sosne from Vertex. Can you hear me okay without the reverb? Hearing me once is bad enough. Twice would be awful. I just, on the slides with the HBs, what was the y-axis? Just trying to understand the scale and the variability. So what, in the HB cells, what was that chloride current or was that protein level?
Yeah, sorry, I didn't explain. This is the flipper assay, so it's fluorescence. Yeah.
Left
Yeah,
here?
I'll
Using chamber is also being carried out.
I'll just yell it so we don't have the
Great.
reverb.
Yeah.
So you showed some really robust data on your screen for these compounds to read through. I was curious, the amount of toxicity that we're seeing in cells, how you expect that to translate in a mouse, for instance, or I guess even in the clinic at that point.
Yep.
And whether or not you thought about doing some experiments in mice to look at distribution as well.
Yeah, that's actually a very good point. I mean, we found it interesting that the most effective molecule was also the most toxic. So what we are now doing with the chemists before actually moving into animals is to change the chemistry to avoid toxicity, but keep or possibly enhance the effect. easier said than done, of course.
So in that regard, I guess my one concern is that you're basically creating a better NMD inhibitor, which you don't want to be a pan NMD inhibitor, but you want to be more selective, right?
Exactly.
So any ideas on how to change the screen to kind of do that?
Well, chemical modifications, Hester groups, yeah.
Okay.
I was a chemist once, but not anymore.
I'll also avoid the mic. Hi, so I'm an organic chemist by practice. So I was really interested on the structures, and actually, because I went to an earlier talk where some of those molecular structures were being used as ion transporters. So chloride ion transporters, did you take that into account when running the assays, or were you able to differentiate the PTC inhibition activity over the chloride transporting activity?
Are you inquiring about its off-target effects on other ion
Yeah, or any
transporters?
effects. So you did some organoid swelling testing, right? So I imagine if the drug itself is moving chloride ions,
Yeah.
it would be interesting.
Yeah, sure. You can argue that about function, but our assays are not functional based. They are based on those tags. So what we look for is inhibition of NMD through the cherry and read-through through the GFP. So when we go back to the cells and to analyze the function, this comment could be, of course, appropriate, but we have to show that the regulation, the CFTR regulation, inhibitors, stimulators, and so on, is actually in place and not some unspecific effect. Yeah.
I see. For the
Swelling, if it's another channel, would it show?
Right. So for the swelling in the organoids, that was also your question?
yeah.
Yeah, so for the swelling in the organoids is actually pretty much CFTR dependent.
I see. So another molecule moving in the ions wouldn't necessarily induce any, I guess, adverse positive response?
Yeah, it's forskolin.
He's inquiring if it's activating another transporter in organoids, and you're just being fooled by swelling. But no, you're doing forskolin.
No. This is the forskolin-induced swelling is very CFTR specific and totally disappears in the presence of inhibitor 172.
I see. Thank you.
Yeah.
Good teamwork here. One more question?
Yes.
I think it
Jason
would be
from
better
UAB.
if you come in front so that we can hear the question, and we'll be happy to repeat it for the audience, because there's some kind of echo happening and we are not getting the question that clearly. And in the meantime, there are people online from patient advocacy group. So for them, if they have not heard the question, the gist of all this is that people have asked is about the toxicity and what effect it can have on other channels. And what Dr. Amaral has said is that she's going to improve the chemistry of these compounds so that toxicity can be lowered. In terms of the specificity of the function she's getting in different cell types, she feels it is attributed to CFTR.
Good.
All right.
I'm from UAB. My question is really small molecule fragments will be effect through. So my question is, if you combine with the peroxide with this inhibitor, and also the other question is DLOX2 was pure. So the thing is, DLOX2, the glucose concentration in lung is too low. So if we have a good way to deliver DLOX2 to glucose, because as we know, lots of delivery methods are developing. So whether that can be bring back combined with this molecule.
Yeah. So the question,
Yeah, if you can
repeat the question. So the question was whether perhaps we could combine some of these compounds with aminoglycosides. That was the first one. And indeed we have. So G418, we do see for the 12 compounds also additive effects. So whatever the mechanism, which we have no idea at this point, is different from G418. they are additive. And the second point was about ELOCs. So we are also testing, I mean, I had the data, but I didn't focus on it. We also, in parallel, also testing with ELOCs to see whether it's the same mechanism of action, different one, and regarding the delivery, that's a big issue. At this stage, we're not addressing it. But it's a good point. Thank you.
Thank you very much. Let's thank Margarita again for a great talk. All right, next up, if you want to come up, we have Batshiva Karem from Hebrew University in Israel. And she'll be talking about ASO therapies when protein is not made. You know what to do here? Click on here. Start.
Good afternoon. Thank you for coming. So I would like to thank the organizers for inviting me to share our progress in the project. And my disclosures are that I am the Chief Scientific Officer and the scientific founder of SpliceSense, and I will be sharing results from SpliceSense. So, as we all know, about 5%, maybe a bit higher, of people with CF have no effective treatments with the currently available CFTR modulators. And these patients include, among these patients are patients that are harboring mutations, nonsense mutations, that do not produce proteins, as we heard from Margarita. And for those nonsense mutations, there is an essential need to develop new strategies, therapeutic strategies. And I would like today to focus on one such strategy and one such mutation, which is the W1282X mutation. It's the second most frequent nonsense mutations in CF. Many years ago, in my group, we identified the mutation and found that it is the major mutation among CF patients from Ashkenazi origins that reside in Israel or elsewhere in the world. And we could show that these patients suffer from a severe disease. More recently, data analysis based on ECFS patient registry database performed by Isabelle Sermet, Eitan Kerem, and others actually showed that people homozygote for premature termination codons, which is a synonym for nonsense mutation or stop mutation, they have a significantly faster, unfortunately, FEV rate of decline compared, here in blue, compared to homozygote F508del. And this was apparent already in younger patients, younger than 18 years of age. They have also shown that the homozygote for a PTC have significantly lower survival probability. We can see the difference in the blue line versus the black line. So we are discussing a severe CF disease that these patients have. So after the explanation that Margarita gave on NMD, there is no need to go into details, but in order to explain our approach, I will just say that, as we all know, each gene at the end has a stop codon that normally signal a stop for protein synthesis. However, a mutation along the sequence can generate a premature termination codon. And this theoretically should result in a shorter protein that probably has lost its function or in some cases can gain deleterious function. But, as Margarita nicely explained, the cells have an RNA quality control mechanism that detects and degrades transcripts carrying a nonsense mutation. And by this, the production of a shorter truncated protein is inhibited. So, as I said, we are working on a specific nonsense mutation, W1282X. The mutation resides in exon 23. And as can be seen here on a Western blot, we don't see any CFTR production. And as a consequence, there is no CFTR function measured in Using chamber. So SpliceSense solution is suggesting a novel antisense oligonucleotide-based therapy for the W1282X mutation. And the idea is to skip over exon 23 where the mutation resides. So what is the rationale for skipping over exon 23? What can we gain from this? So the first consideration is that skipping over exon 23 will bypass the nonsense-mediated decay because it will skip over the mutation. And thus, transcripts carrying W1282X are predicted to result in messenger RNA, stable, and such messenger RNA can be translated into proteins, shorter ones that are deleted for the skipped exon. The second point relates to the location of the mutation in the CFTR protein. So exon 23, where W1282X resides, is part of the second nucleotide binding domain. And it was shown previously by Jacques Riordan, Christine Baer, and others that proteins that are lacking the entire NBD2 and the region which is downstream of it retain partial CFTR activity that can be augmented by CFTR modulators. And the third point is that the exon is in frame and therefore can be removed without causing a frame shift in the open reading frame. So this is the basis for our idea to skip over exon 23 as a potential therapeutic approach. And of course, the question is, will CFTR proteins lacking exon 23 be functional? Okay, so our suggestion is to use, for skipping on exon 23, is using antisense oligonucleotides, which are single-stranded RNA-like short sequences that can bind by base pairing to the RNA and modulate the splicing of the target. ASO are specific. They are designed to be specific and thus theoretically avoid off-target effects. They are chemically modified to enhance stability and increase cell uptake. And they are clinically relevant. As of today, there are 12 clinically approved antisense oligonucleotides on the market, and several of them are modulating exon retention, so they are splice-switching ASOs. In SpliceSense, we developed an in-house algorithm to design antisense oligonucleotides. It is a very nice tool which can identify splicing motifs within the targeted sequence, which it can design ASOs that are efficiently and specifically bind the target sequence, and also we take into consideration immunogenicity optimizations of the ASO. So for this project, we designed about 80 consecutive antisense oligonucleotides. And the first step was to analyze the effect of exposing cells to these ASOs on the splicing pattern. Do they promote exon 23 skipping? So we can see here two approaches, real-time PCR and RT-PCR, and in both of them, we can see that some of the designed ASO have a very significant effect. In the real-time PCR, we can see that some of the ASO leads to a very, very low level of exon 23 retention, so most of the transcript, exon 23 was skipped. It is easy to see it in the RT-PCR, in these gels. We see that with control ASO, all RNA includes exon 23 as expected, and here are three different ASOs that almost completely skipped over exon 23. Of course, next was the important question. Can these transcripts be translated to a protein? Are they stable and can be translated? And the answer is yes. We can see here a Western blot. Following treatment with a control ASO, there is no protein, as we saw earlier. Using one of these three ASOs as an example, we can see a significant level of CFTR protein. Mature CFTR protein here is Ben-C, although it is deleted for exon 23. So the ASO 23-2 was the one that we chose to go on with, so it will be called from now SPL 23. And the next question, of course, is, are these ASOs functional? Can proteins that were generated following the treatment, are they functional? So in this case, we switched to human nasal epithelial cells. We treated, human nasal epithelial cells from 3 patients homozygote for W1282X and 5 patients, or H&Es from 5 patients heterozygote for the mutation. And we measured the CFTR function by Using chamber following the exposure to the ASO plus ETI. As we said, this can be enhanced by the ETI. So we can see here in homozygote a very nice effect in all 3 homozygote cells. In one of them, the function reached almost 40% of wild type, while the others were somewhere between 15 and 20% of wild type activity. Among the heterozygote patient, now we are targeting 1 allele. We can see that in 1 patient, the response was quite low, but in the other 4, we reached 10% and even higher, with 1 cells reaching 25% of wild type CFTR. So our SPL23 indeed led to a significant increase in CFTR activity. And the last experiment in this set of experiments was to test whether the antisense oligonucleotides are specific for the W1282X. And as can be seen here, the answer is yes. In cells from healthy volunteers, they are not carrying the W mutation. The skipping was minimal. And thus, the function did not change as expected. And this is important for heterozygote patients carrying the W1282X on 1 allele and another mutation on the other allele in order not to skip over exon 23 in the other allele. We don't know what the future will be, if this allele will have any targeted therapy, etc. So we can conclude that SPL23 leads to a minimal effect on exon 23 skipping, which does not affect CFTR function in human nasal epithelial cells from healthy volunteers. To summarize this part, we have seen that SPL23 promotes high level of exon 23 skip transcript, promotes production of mature CFTR protein, and this by itself indicates that the exon skipping is indeed overcoming the RNA degradation by the NMD mechanism, which is the major obstacle preventing the effect of modulator drugs on the W1252X allele. So by this, using this approach, we don't need NMD inhibitors or readthrough agents as we bypass the problem by skipping over the exon. We also showed that there is a significantly restoration of CFTR function together with ETI, and that potentially this can be translated to a clinical benefit for patients. So splicing's end products are inhaled drugs. So in this audience, we don't need to elaborate much on the advantages of inhaled medicines. I just want to emphasize that by inhalation, inhaled drugs, we minimize systemic exposure. And in the case of antisense oligonucleotides, this is really important, as systemic exposure of antisense oligonucleotide can be toxic. Patients can develop thrombocytopenia and other symptoms. And another point that I want to emphasize is splicing's end products are given to patients once a week, thus reducing dramatically patients' treatment burden. Next question was, can ASO penetrate the mucus within the lungs, enter the cells, and access their nuclei? I will not go into too many details because of lack of time. I just want to emphasize few results. So as I emphasized earlier, ASOs are single-stranded and very small molecules. They have the size of 1 to 3 nanometer, and they are significantly smaller than the mucus pore size, which is in healthy individual 100 nanometer, in COPD patients 50 nanometers, so they are tiny, and they should be able to go through the mucus pore. They are negatively charged, and the mucins that construct the mucus are also negatively charged, so theoretically they should repel from each other and go through. And in a very elegant series of experiments that I will not show today, we showed that the ASO can penetrate through the mucus that is generated in HBE, in differentiated HBE cells. What I would like to show is experimental results from animals that were inhaled or enforced to inhale the ASOs. So first mice, wild-type mice, but also mice, beta-ENaC mice, that they are a model for muco-obstruction. They have CF-like mucus. And in both cases, in both type of mice, we can see a wide distribution, an efficient distribution of the ASO in the lung and in all areas of the lung, from the trachea, the bronchi, down to the alveoli, and same results we obtained in non-human primates. Again, uniform, efficient distribution of the ASO in all the parts of the lung. So, of course, the next question is, does the ASO-based therapeutic approach have a potential to provide an effective treatment for people with CF carrying the W1282X mutation? We don't have, at the moment, an answer to this since we did not initiate yet a clinical trial. But I would like to share with you new results from another project in splicing targeting another CFTR mutation. I will describe this project tomorrow in Symposia 16, but I would like to show the results as a validation of the platform, as a validation of Splicen's ASO platform. So without going into any details, with the mutation, the splicing mutation that we are targeting with an ASO, we reached the clinic. So we completed successfully phase 1, which was a single ascending dose in healthy volunteers. And the results showed no significant adverse event and no significant related adverse event, thus showing promising safety profile. And there was the systemic exposure to this ASO was low and dose dependent. We already initiated phase 2, which was a placebo-controlled multiple ascending dose in subject carrying the splicing mutation. And the aim of the study, the aims were first to evaluate safety and tolerability of the multiple ascending doses, and secondary, to assess the preliminary efficacy by FEV1, lung disease, of the multiple ascending doses. And earlier this month, Splicence announced positive results from our ongoing phase 2 study. These are interim results, preliminary. We are analyzing the data and soon we will publicly show them. And the key highlights are that there is a strong safety profile from the phase 2 that we already, the result that we already have, with no safety signals identified so far. And there is meaningful efficacy. There was lung function improvement observed in up to 70% of the patient, with a mean absolute FEV1 increase of 10% compared to placebo. And if we look at the individual patients, for each patient, the threshold was at least improvement of 5% from its baseline FEV1. So as far as we are aware of, this is the first evidence of potential clinical benefit of ASO therapy in a pulmonary disease. And as I said earlier, this is a pipeline validation. The data support and encourage us to advance additional inhaled ASO programs into the clinic, and this includes W1282X that I hope soon we will be able to move into the clinic. And here is the pipeline of Splicence. So beside the splicing mutation, W1282X, we have other projects aiming to target mucins for other pulmonary diseases, including CF. And here are the poster numbers in which these results are presented. So I would like to finish my talk by thanking my lab. So this is my current lab, and here are the people along the years that contributed to what I presented today. And here is Splicing's team, Efrat and Lital, I hope they are here. So we are very proud of Splicing's team. And last but not least, our collaborators, Isabelle Sermet, Martin Menz, Christine Baer, Steve Rowe, and Eitan Kerem, who is involved in every part of the project. And here are the funding. And thank you for listening.
Questions?
Yeah. I was wondering if you could clarify how the ASO you were showing is specific to the W1282X and not marginally affecting a different CFTR allele splicing.
It's the location of the antisense oligonucleotide. They are so short that any mismatch is hindering their ability to stably hybridize. So That's the answer.
So essentially that's targeting the W1282X mutation itself, is what you're saying.
Could hear you, sorry.
It's the ASO overlaps.
I think it's.
Yeah, the answer is yes.
Yeah, that would be shared by the other allele, right?
Yeah.
Right?
Yeah.
That's what I'm getting at.
Yeah, the answer is yes, if I understood correctly your question, yeah.
So it's specific to that
Yeah, it's
mutation
specific
only.
to the mutation, yeah. So it's very specific. The disadvantage is that it can target only this mutation, but as I showed, it probably does it very
So
effectively.
just to clarify, it's binding that SNP.
It's the single nucleotide change
Correct.
makes it a nonsense mutation. The ASO is binding over that sequence. So you have a one nucleotide difference between the wild type and W1282.
Okay, all right. That's all I wanted to know. Thank you.
Ashley?
Wonderful talk. You've shown very impressive data with how well these ASOs can be taken up by the cell. Is it possible to conjugate it to another cargo, perhaps to use that as a method for whether you use it therapeutically or to learn something biologically?
Could you hear anything? I'm afraid that because of the echo, we cannot hear.
Can you conjugate the ASO to another cargo?
About cargo, no. This is free uptake. It is given to the cells with no cargo. They are so tiny. And this is a big, big advantage. You just expose the cells to the ASO and slowly they penetrate through the endosome. They travel from the endosome through the cytoplasm into the nuclei, right, where splicing occurs. So, yeah.
Could it be conjugated?
Could it be conjugated?
To improve delivery.
Yeah. At the moment, we think that there is no need to conjugate these ASOs.
Thank you.
Yeah, that was my question. Hello. It's naked ASO. Naked.
Yeah, it's naked ASOs, as I explained. It's just given to the cells or to the patients in the inhalation, of course.
This question is just maybe stemming from other clinical trials where they've conjugated Fabs and antibodies like dyne and avidity that have been used in skeletal muscle. Yeah.
Thank you. I have a follow-up question. For the carrier-free oligo, can you clarify the modification, the chemical modification on the ASO?
Chemical modifications.
To make them stable.
So what is the question?
What are they? And if you do them, and you probably aren't going to share what they are, but like,
Yeah.
they modify.
Yeah, they are modified,
Yeah.
as I said, in order to increase both the cell uptake and the stability. So they are chemically modified.
Yeah. What are the modifications, if you can clarify? No.
Okay, we can discuss it
Thank you.
after that.
Hi. I was very impressed by the data on the 3849 oligo showing an increase by 10% in FEV1. So this variant is also responsive to ETI. So my question, did you test for the additive effect of the ASO plus ETI in these patients?
I'm not sure.
Did you do an additive effect with ETI?
And the ASO?
Yeah, ETI plus
Because it's responsive.
All the experiments are exposure to the ASO plus ETI.
No, but also in patients in the clinical trial?
No, he wants to know if you're going to do it in the clinical trial.
The answer is probably no. We will start with W1282X possible responders, homozygote, or, yeah. Let's see that we can make it first before we will perform more complicated.
So regarding the data on the 12...
Okay. Yes, please.
Regarding the data on the W1282X, the variability that you observe between the different individuals, does that correlate with the basal levels of NMD and of CFTR RNA?
Does the amount of activity you get correlate with the amount of basal level you get of the RNA? So when you saw patient-to-patient variability, I think is what he's asking.
Yeah. I have no good answer to your question whether we have a correlation with the RNA that we can stabilize. That's something that we are looking into. So what Ethan came to advise is that I will say what I will show actually tomorrow with the 3849 project, that there we aim for combination therapy. But tomorrow it will be more clear what do I mean by this. Thank you, Ethan.
I have a question. So the patient-to-patient variability was kind of striking. Did this inform on your clinical trials and how many individuals you would need?
Yeah. Because of the variability, we probably will need more patients. Okay, as I will describe tomorrow for the 3849 project, we started with very small number of patients just to see whether there is any chance to see something. And the variability is a problem.
Yeah.
Yeah. Okay, so must be more patients.
Everybody needs a good statistician. Any other questions? If not, let's thank Batchiva for a great talk.
Thank you.
All right. Next up, we have Andrei Korostolev from University of Massachusetts Chan Medical School in the U.S. And his talk is titled Read-through of CFTR Nonsense Codons by R ASOs. Take it away.
Hi. I'm going to talk about a new modality of antisense oligonucleotides that my lab has recently discovered, and it was great to hear Batchiva's talk about this approach, antisense therapy in general, as they're leading the way in figuring out how the delivery can be achieved, and this is very important. So we're now starting this on this path, and we think about a different approach, which I'd like to present from a structural biology perspective, because my lab is interested in structural biology and understanding the mechanisms of translation in general, and that's what led us to designing this approach to the read-through of nonsense codons. I have no conflict of interest. So, a read-through of premature stop codons, as you have heard about many times already, depends on this balance between termination on the premature stop codon. So termination results in the release of an incomplete protein that is not effective, and elongation, which involves decoding of the stop codon by tRNA. And in most cases, except for suppressor tRNAs, this process of elongation would be inefficient because there's no tRNA that could decode the stop codon. So only tRNAs which have two nucleotide matches could do that. So this process is inefficient. Nevertheless, a very small portion of mRNAs do undergo read-through, resulting in the production of full-length protein, which is very inefficient. So our goal is to either reduce termination or increase elongation by either introducing tRNAs that can read the stop codons, or by other means to achieve to shift the balance from termination to elongation. And there are various approaches that you have heard from in this conference and from other sources, including downregulation by using inhibitors of release factors that recognize the stop codons, or to enhance elongation by using suppressor tRNAs that specifically can recognize nonsense codons, also called premature termination codons, or using small molecules such as aminoglycosides that can bind to the ribosome and tweak it into decoding codons by using a tRNA. Now, one problem with most of these approaches is that they do not distinguish the specific premature termination codon on the specific mRNA that causes the disease from most other mRNAs. So there's a possibility of reading through normal termination codons throughout the cell, and that might result in toxic side effects, especially following the long-term application of these approaches. So I've been long interested in how we could induce mRNA-specific read-through. So I'd like to share some studies that we performed before arriving to this idea that are really focused on understanding the detailed mechanism of decoding by the tRNA, and also I will discuss the mechanisms of termination where the codon is decoded by proteins called release factors. So, as many of you know, the tRNA is bound with elongation factor, which is a GTPase, and it's brought to the ribosome, to the ribosomal A site. And when the codon matches the anti-codon of the tRNA, GTP is hydrolyzed and elongation factor dissociates with GDP so that the tRNA can be incorporated into the A site and present its amino acid next to the peptidyl tRNA where the peptide chain is growing. And importantly, there's a sarsen-ricin loop on the large ribosomal RNA that is involved in catalyzing GTP hydrolysis. Now, if tRNA is not matching the codon, then normally it gets dissociated at either the first stage, which happens prior to GTP hydrolysis, or after that. So the main questions are, how does the ribosome select the correct tRNA based on this very slight differences between the matched and mismatched nucleotides, which were shown previously to be energetically insufficient to determine this high rate of decoding accuracy. And the second question is, how does the ribosome communicate between the decoding center and sarsen-ricin loop to achieve this correlation of decoding, accurate decoding with GTP hydrolysis. So we used time-resolved cryo-electron microscopy, which allows to achieve near atomic resolution of the ribosome with tRNAs and other factors. And here is the approach we used. We actually performed the reaction and collected data points or data from several time points so that we could restore which fractions of the ribosomes are involved in decoding the mRNA. And here I'm showing you our calculated proportions of the functional states of the ribosomes, which, as you can see, look very much like a time progress curve of a biochemical reaction, which was really great to see in that we can calculate that certain ribosomes perform the accommodation of the tRNA, so they turn into products, and the number of ribosomes that are substrates, meaning they have the empty A site, they slowly decrease with time. And then, as you can see, we also have a very small, rise in the ribosomes bound with elongation factor TU, and then this ribosome fraction decreases as expected for an intermediate. So I'm not going to go into details of every structure. I just want to show that we can capture individual states of EFTU intermediates through the process of decoding, then tRNA accommodation, peptidyl transfer, and the resolution that we can achieve allows us to distinguish details such as the presence of triphosphate on GTP, and of course we have detailed information about what's happening in the decoding center. So with that, I'll summarize what have we learned from doing cryo-EM. And we actually compared how the ribosome interacts with the near-cognate tRNA and cognate tRNA. And we found key differences, that the ribosome undergoes a conformational change that is absolutely essential for it to make the decision, so to say, whether to accept the tRNA or to reject it. So when the near-cognate tRNA binds the A site, as you notice, we I show the actual structures here, there was no difference in the conformation of the ribosome. So the tRNA binds to the A site, interacts with the codon, and dissociates because it inefficiently forms these base pairing interactions. However, when the cognate tRNA binds, it induces large conformational changes in the small ribosomal subunit. So the ribosomal subunit the small subunit consists of a shoulder domain that is labeled here, and it's kind of closer toward us. And in the back, there's a body domain. So you will see that the shoulder moves when the cognate tRNA binds. So I'm going to show this again. And this change, although it seems subtle when we look at the whole ribosome, it is actually pretty large, and it's very important. Now, to give you an impression of what we can see, what kind of level of detail we can observe, I'm showing the decoding center and conformational changes that result in that conformational change. So the local changes allow the shoulder domain to move towards the body only when the full base pairing, full Watson-Crick helix is formed through base pairing. So when the near cognate tRNA binds, it fails to induce conformational changes in the decoding center, and tRNA can dissociate. However, when the cognate tRNA binds, a full Watson-Crick helix is formed, and now one of the nucleotides on the shoulder flips and interacts with the Watson-Crick helix and moves toward the body domain. Now, why is that important? It is important because the shoulder is where the GTPase binds, and it binds initially far from this catalytic sarsen-ricin loop. So the shoulder domain is necessary to move the GTPase closer to the sarsen-ricin loop and induce the hydrolysis of GTP. So we can see here how a catalytic histidine interacts with the sarsen-ricin loop only when the shoulder domain moved it closer. So we were able to reconstitute this reaction and see the details, so we can now appreciate how these subtle differences actually affect this large ribosome structure. And it also allowed us to take another look at aminoglycoside antibiotics that were previously published, structures of which were previously published by the Ramakrishnan and other labs. So what they and other labs have found is that aminoglycosides bind very close to those nucleotides that change conformations in the decoding center, and they force them to interact with tRNA even when there's a mismatch. And when that interaction happens, the shoulder moves closer, and that induces GTP hydrolysis, so the tRNA gets accepted. Now, that also explains why aminoglycosides are so toxic. The problem is that, of course, they do not distinguish between stop codons or sense codons, because the nucleotides that I'm talking about in the decoding center, they only face the backside, so to say, of the base pairing. They do not interact with the nucleotide bases, so they cannot distinguish between, stop codon or a sense codon. Okay, so how termination differs from decoding by tRNA. As I mentioned, termination happens when proteins called release factors, namely ERF1, which acts somewhat similarly to tRNA, and ERF3, which is somewhat similar to FTU, are delivered to the ribosome. And another very important difference is that stop codons in eukaryote actually also includes the subsequent nucleotides. So it's not 3, but 4 nucleotides that are recognized by release factors. So here are the structures that were determined by the Ramakrishnan and Beckman labs that show how the conformational changes in release factors result in the change in RF1 and release of the peptide from the ribosome. So as you can see here, delivery of elongation of release factor 1 is somewhat similar to EFTU and tRNA as I showed before. When GTP hydrolysis happens on ERF3, upon recognition of the stop codon, ERF3 dissociates and the release factor changes conformation, inserts into the peptidyl transferase center, and hydrolyzes the linkage between tRNA and the peptide. So that's what releases the peptide. And of course, this process is irreversible. There's no way to link them together. So that's what we're trying to prevent on the nonsense codons. Now, as I said, what's important to know is that there's the fourth nucleotide after the stop codon that plays a huge role. And that was discovered 30 years ago by Warren Tate lab, who reported that efficiency of translation termination is dependent on the base following the stop codon. So what they found, both in vivo and in vitro, is that if there's a purine that follows the stop codon, then termination is very efficient. However, if there's a pyrimidine, termination is less efficient. And this range of efficiency is very broad, almost 100-fold, depending on which nucleotide is after the stop codon. So structurally, we can confirm these biochemical findings by looking at the interactions of the stop codon. So what you can see here is that there are 4 nucleotides that are kind of cramped into the A site, and the 4th nucleotide interacts with the same nucleotide that I showed previously that flips when the tRNA is recognizing the normal codon. So here we have 4 nucleotides packed into the A site instead of 3. So Denis Susorov in my lab and I were thinking, can we capitalize on the structural difference between termination and elongation? So as schematically is shown here, release factor kind of pulls mRNA into the tunnel by 1 nucleotide, which does not happen with tRNAs. So our hypothesis was that we can make sequence-specific read-through if we introduce an oligonucleotide that would bind downstream the nonsense codon and interact with the ribosome in such a way as the ribosome moves through the mRNA so that it prevents that pulling of the mRNA into the tunnel. And if that works, then it means that the release factors will not be able to bind to the compacted stop codon, and termination would be inhibited, and there will be more chance for the tRNA to read the stop codon and elongate. So we started by making constructs which contain the G542X mutation, which, as you know, is the prevalent nonsense mutation in CFTR patients. And we included a large region of CFTR that contains most of the sequence context that is important for the efficiency of elongation or termination. And we didn't include the whole gene because we also needed to have a reporter, which is in our case nanoluciferase. So nanoluciferase follows the nonsense codon, and we would see signal if we induce translation in a mammalian cell lysate system, which is a great system to do assays that require a lot of screening, which was our case. So if the ribosome reads through, then the full-length nanoluciferase is produced, and we would see light. We started with making 3 constructs. One was the actual nonsense mutation, which unfortunately is followed by guanosine, which makes it a strong stop codon, so it's harder to read through. Now, for the purpose of optimizing our screen, we also made a mutation following the stop codon, which is a C, that is easier to read through, so that would be a good indication for us that our approaches might be working. And then we also made the wild-type glycine construct, to which we could compare the read-through efficiency. So as expected, when we just throw the construct into a translation system, what we see is that there's plenty of the normal sequence production with the sense codon. However, UGAC and UGAG do not make protein efficiently, and UGAC makes a little bit more as expected because termination is less efficient. And that is confirmed by Westerns, so you really don't see much of a protein made except when there is a sense codon. So we then started screening various positions by using oligonucleotides, and we started with using DNA oligos because this translation system is rather forgiving, and it allowed us to actually identify the optimal position. So we changed the composition of the oligonucleotide position relative to the stop codon, the length, and a lot of other parameters. And we found that consistently we do observe read-through. That was really exciting to see, indicating that it's a very specific mechanism. It happens only when the oligonucleotide is placed 5 nucleotides downstream of the stop codon, or we also call it the +8 position if you name the U of the stop codon as number 1. So we then repeated that with some other sequences and observed that consistently you do observe read-through when the oligonucleotide is placed 5 nucleotides downstream. This is confirmed by the Westerns. So we then looked at the nonsense codon that is more challenging to read through. And of course, it turned out that it is challenging to read through for most approaches, including antisense oligonucleotides. So we started looking at combinations of oligonucleotides with other read-through agents at lower concentrations. So the idea here was that if there's synergy between these different approaches, then we could possibly induce read-through without causing that much toxicity by those less specific compounds. And so we tested antisense oligonucleotides with the actual nonsense codon on G542X sequence, in combination with G418 or with suppressor tRNA that can read this codon. So what you can see here, antisense oligonucleotide induces very mild, very low amount of read-through, barely visible compared to the background. The same is true for lower concentrations of the G418 aminoglycoside and suppressor tRNA, again, at the concentrations that we used. So you do not see any read-through when you use them alone. However, when we start combining, the effect is synergistic, which was really exciting for this challenging mutation. Both G418 and ASO and tRNA and ASO result in read-through that now is probably about 20% or so of the wild-type protein. And that, again, is confirmed by the protein Westerns. So we then asked whether the mechanism by which it works actually is what we thought. Does oligonucleotide actually induce inhibition of translation termination? So we developed a different assay which allows us to purify ribosomes sitting at the end of the nanoluciferase mRNA, and we can add in a controllable manner release factors with or without any other compounds and see if they result in the release of nanoluciferase from the ribosome. And we found that this results in the proper folding of nanoluciferase and the nanoluciferase signal. So what we find is that when we bind, when we add release factors with just the buffer or with oligonucleotides that bind to some other positions, we do see some range of termination efficiency, which is still pretty efficient. But when we add the oligonucleotide that specifically binds to position +8, we do see the decrease, as you can see from this curve, of the release of nanoluciferase, indicating that indeed it inhibits termination. Now, as I said, we started with DNA oligonucleotides, but those are not what you want to deliver into cells. So our next step was to optimize them by chemical modifications. And unfortunately, we found that although we can modify most of the oligonucleotide, there are some challenges which I will mention later. Anyway, we collaborated with my colleague at the RNA Therapeutics Institute, Anastasia Kvorova, who runs a really large core where they can produce a lot of different modifications, some of which are listed here, but these are not all. So standard ones are 2'-O-methyl, 2'-fluoro, phosphorothioate, and some others. And as you can see here, we could achieve read-through when we modified most of the oligo, but not all. So here are the data showing that modified oligonucleotides are as efficient, if not slightly more efficient, than the non-protected oligonucleotides. That's shown in the protein gels. Now, importantly, because DNA binding also causes RNase H-induced cleavage of the mRNA, which we observed in our translational system, when we modify the oligonucleotide so that it doesn't resemble DNA anymore, we do see that the mRNA becomes much more stable, as you can see in the third lane here. So that was very promising and exciting. Nevertheless, only recently we were able to find the pattern of modifications that allowed us to retain full activity and make the oligonucleotide fully modified, which we're really happy about. Okay, so I will conclude here by saying that we found that there is a possibility of inducing sequence-specific read-through by using antisense oligonucleotides, which we call read-through inducing ASOs or RASOs, by inhibiting termination. Combination with other read-through agents at lower concentrations are synergistic. Modifications do increase mRNA stability and read-through efficiency, and I'm happy that we now have 100% modified RASOs. We confirmed on 5 additional disease-causing sequences that RASO approach is working, and now there are in our lab studies underway where we're testing these in cell models, and we're also we have produced constructs that would test W1282X and other prevalent or less prevalent mutations in CF patients. With that, I would like to thank my lab and the cryo facility for the studies that I showed in the beginning, my collaborators Anastasia Kvorova and Nico Grigoriev, and funding from the NIH, Cystic Fibrosis Foundation, and Emily's entourage. Thank you for your attention.
Questions? I have some.
I thought you would.
Yeah. So it's really interesting about the small changes that you see in the ribosome having, like shifts in structure, having huge changes, effects on read-through. So if you looked at changes in anticodon loop tRNA modifications and how that would affect this mechanism?
We haven't done this, but there's a body of literature showing how tRNA modifications do indeed affect the decoding process, and it's known pretty well for example, the most notable, well, one of the most notable modifications is the modification of the nucleotide that follows the anticodon, and usually that nucleotide is modified in such a way that there's a huge, chemical moiety that's attached to that nucleotide. What it does, it stacks on top of the 3 base pairs of the codon-anticodon helix as a pretty much 4th base pair. So that stabilizes this very metastable 3 base pair helix. So that's one example of how modifications do affect decoding, but there are other mutations in the anticodon stem loop and other positions of tRNA that also affect the dynamics of interactions between tRNA and the ribosome. So they are very important.
And a follow-up question is, have you thought to use the drug DAP and convert tryptophan to a suppressor for UGA in accordance with your RAS?
That's a great idea, and we are testing combinations with other compounds. Of course, we are thinking about including this small molecule as well, but we're also looking at using those release factor degraders, NMD inhibitors, and other small molecules.
Dave?
Yeah, Andre. Nice talk. So your oligos are sitting, you said 5 nucleotides. They start 5 nucleotides downstream of the stop codon, correct?
That's correct.
So is this a steric hindrance that's pausing the ribosome, or is it would be in the mRNA channel still, right?
Yeah, so based on, there is no structure yet, we're working on it, of a duplex that would be sitting at the mRNA entrance. But you're pointing in the right direction that based on bacterial ribosome structures with that aimed at understanding the helicase mechanism. So the ribosome is a very efficient helicase. It is, though, the elongation process that drives, this unfolding of RNA structure as the ribosome plows through. So it is at the entry into the mRNA tunnel where position +8 is, interacts with the ribosome. Now, we don't have data to show what exactly in that position interacts with the helix, but it's very interesting that it does affect termination, probably through the mechanism that I described and how we hypothesized. But of course, it's not surprising that it doesn't affect elongation at all. We did test it on, various positions because the ribosome just, strips it off during normal decoding by tRNA and subsequent translocation.
If it really is just the base pairing that's causing the problem, could you just introduce a stem loop in the mRNA, and if you saw the same effect, would that confirm it?
Right. There are examples of structures in mRNA that actually do affect the efficiency of resources that in part, inspired us to think about this strategy. So yes, there are examples in nature of downstream structures.
Bachila.
So 5 nucleotides. We tested in G508del, tested for ligands as well, seeing it's general.
Yes.
Can you repeat the question?
Yes, the question was whether positioning of the antisense oligonucleotide, 5 nucleotides downstream the stop codon, is uniformly acting on other nonsense mutations as well. And my answer is yes, that's what we're finding on other stop codons.
A question. So in short, can you explain to the audience that when you're studying this fascinating machinery, what is your construct here? Because you are addressing a problem of a nonsense variant. What are you studying? And when you're studying the translation, is it a reticulocyte lysate translation, or is it in some cell-based system that you are getting the output from?
So we've been using the commercially available rabbit reticulocyte lysate, but now we switched to the mammalian lysates, which we're preparing. So these are just human cell lysates that act in the same way. However, they have a little bit more to them, so they more accurately represent what happens in the cell. And we do see that using fully modified oligos works as efficiently and does not degrade mRNA and indeed, induce a spike in protein production.
So you're not recapitulating any glycosylation patterns then, if you're using reticulocyte lysate system?
Well, as I said, we also use mammalian cell system, but we haven't tested for post-translational modifications.
Thank you.
Great. All right. Let's thank Andre for a great talk.
Thank you very much.
All right. So next up, we have Sriram Vaidyanathan from Nationwide Children's Hospital, Ohio, USA. His talk is titled Strategies to Replace the Full-Length CFTR cDNA in Airway Stem Cells. Take it away.
Okay. Good afternoon, everybody, and thanks to Neeraj and John and the organizers for inviting me to present our work here. So I don't have any disclosures, and I would just like to start with acknowledgements. We're a new lab. These are all the lab members that are working hard on all the experiments I'm going to describe to you soon, and I would like to thank all the collaborators and sources of support. And I don't need to reintroduce this topic. I think all the previous speakers have really beautifully explained why we need more alternatives, especially for people who do not benefit from modulator therapies. But the one point I wanted to explain a little bit more about, and in order to motivate why we're looking at CFTR cDNA replacement, is the question of whether there are many people that are ineligible for modulator therapies and how the spread of different variants might sort of inform which parts of the gene or whether full cDNA replacement is better. And so to motivate this, I wanted to point to some work from Lily Barr in my lab. She's a former member of my lab, and this manuscript is under revision, so hopefully it's going to come out soon. So we have been looking at CF across different populations to get a sense for how these variants that are non-responsive to modulators might vary across different populations. And we used Nomad and also a private data set to sort of get started on this. And the first question we asked was just the prevalence data, right? And we all have known that CF is more common in European ancestral populations, but then it's rare in other populations. But I think a lot of previous studies have not accounted for differences in births, right? So when you're looking at birth rates across different parts of the world, and when you adjust for that, what we see is that even places like India, China, or Nigeria, where we expect fewer people to be affected on a per 100,000 birth level, when you adjust for the higher birth rates, you end up having the number of people affected by CF end up being comparable to places like the US and the UK. And so then when we look at modulator eligibility in these populations, and so we started, on the left is some work that I had done in my postdoc where we had looked at this question for the Asian population using registry data. And here we had observed that about 40% of Asians from different parts of Asia ended up being ineligible for modulator therapies. And then when we asked the same question in Nomad, here you can't essentially go and look at a number of people, but you can ask the question in terms of what fraction of pathogenic alleles in each population is affected by variants that are ineligible for modulator therapies. And we come to a very similar sort of an answer. About 30 to 40% of pathogenic alleles in a lot of these populations end up being affected by variants that are ineligible for modulator therapies. And it just boils down to the fact that many of these are nonsense or splicing variants. So then that takes us to the question of how these variants might be spread across the gene. And if you've seen my talk before, this is a very old slide, but essentially the point is that these variants that are ineligible for modulator therapies are often spread across different parts of the CFTR gene. And in the bottom is a more quantitative representation of how this might look. And so essentially all of this is just to say that replacing the CFTR cDNA might be a very efficient way to sort of address all people affected by CF that may not respond to modulator therapies. However, it's really hard. So what is the status of CFTR replacement? So we all know about some really interesting messenger RNA and regular gene therapy work where there have been a lot of attempts to either deliver CFTR using AAV or now lipid nanoparticles. And these are what I would call transient gene therapy attempts where people have, or groups have tried to transiently deliver the CFTR cDNA. Of course, the disadvantage here is that the expression goes away and you need redosing. The effort that I'm going to describe to you focuses on genome editing, where the idea is that we replace the CFTR cDNA in airway basal stem cells. And when these stem cells repopulate the airway epithelium, you have durable restoration of CFTR function. And so when we think about genome editing, my lab has been focusing more on the ex vivo genome editing application. So our idea is to take airway basal stem cells, gene correct them ex vivo, and try to transplant the corrected cells back. And this is mostly just because the homologous recombination-based genome editing approaches are much more efficient in dividing cells, and so this allows us to leverage this concept where we're hoping that at some point we can transplant these cells into some parts of the airway, and then this would restore CFTR function and you would get some therapeutic benefit. The proof of concept that you can replace the CFTR cDNA was already published, so I'm just going to briefly gloss over that, and then I'm going to focus most of my talk on enrichment-free insertion of the full CFTR cDNA in airway basal stem cells. So to get us started, the flavor of genome editing that we're using relies on the cell's endogenous machinery. So we use Cas9, a bacterial protein, to induce a double-stranded break in the genome. And then we rely on homologous recombination, where we supply a template that is delivered using AAV. And then the homologous recombination machinery recognizes this template, and then because of homology to the cut site, it can insert your transgene. Okay. The challenge here is that the CFTR cDNA is still too large to fit into an AAV, and so it doesn't allow us enough room to include homology arms that make this process efficient, and then there's really no room for any sort of marker. And so the strategy that we have been using is to split the gene into two. And so we make a break near exon 1, and then we put in the first half of the cDNA, and then we make a break at the end of this first half, and then we put in the second half. And then we added a truncated CD19 cassette to sort of detect these edited cells and enrich for them if necessary. So the corrected locus at the end looks like this, where you have the first half right at exon 1, and then the second half, and then an enrichment cassette. And as you might imagine, this process is super inefficient. So the paper that was published a few years ago, we reported less than 5% corrected cells. It required enrichment with MAX, but once you enriched for the corrected cells and differentiated them, you were able to get non-CF level CFTR restoration. Of course, our interest is to get rid of the CD19 cassette and maybe replace it with some more interesting cargo, especially given the fact that the transplantation part of this project is a little bit more challenging. And so that's why the interest in my group for the past 3 years is to improve this gene replacement to a point where you don't need to enrich the cells. And so when you're thinking about improving CFTR cDNA replacement, you have a variety of options, right? So you could go and explore viruses that have larger packaging limits, or you could go look at non-viral methods or templates that would allow you to replace this CFTR cDNA. And so when we got started, we did attempt to go with larger viruses. So here's an example where we tried to package all of the CFTR cDNA and the enrichment cassette into adenoviruses. And even though we're using a 2 AAV system, compared to that, the adenovirus-based system was much less efficient. We barely got any gene insertion, and we don't know if this is because of innate immune responses elicited by the adenovirus, or if this is because of other structural differences, like the presence of end caps in adenoviral genomes. Karen last year presented some of her attempts to look at single-stranded DNA templates. So in this work, we were asking the question of, so the status quo for single-stranded DNA templates is we modify the ends and we leave the middle unprotected. And so the question we were asking is whether we could modify more nucleotides within the templates to improve gene insertion. And modifying more of the template does improve gene insertion. However, it's still much less efficient than AAV-based templates. So we're looking at other ways to improve single-stranded DNA stability. And one study that had come out when we were thinking about this was the nuclease TREX1, which is thought to degrade single-stranded DNA. And in our studies, what we noticed was that iPSCs do not express TREX1. And compared to the other cells, iPSCs have much more efficient genome editing. And so we wanted to knock out TREX1 from airway stem cells. And you can see that when we knock out TREX1, even unmodified single-stranded DNA can mediate efficient gene insertion in these airway stem cells. So since we cannot make chemically modified long single-stranded DNA at scale yet, we thought maybe we could go and knock out TREX1 from airway stem cells, and that would help us replace the full CFTR cDNA without any enrichment. And so that was something that Reza started. So we got single-stranded DNA that would express GFP. And as you can see, compared to airway stem cells and vascular endothelial cells, iPSCs underwent much more efficient gene insertion. So it fit the paradigm that TREX1 was the culprit in degrading single-stranded DNA. However, to our surprise, when we knocked out TREX1, we did not see any improvement in gene insertion of this gene length template, which was really surprising. And we don't have a good answer for this, right? So we were thinking of different bottlenecks that might affect gene length single-stranded DNA differently from oligonucleotides. And we were also wondering if the gene length templates were undergoing insertion through a different pathway compared to the short oligos. And we did look at that. So there are two different homology-mediated pathways. So one is single-stranded template repair, and then the other one is homologous recombination. And they are mediated by different proteins. And so we started by knocking down these different proteins. And you can see in the Westerns that we were able to knock down these different proteins. However, both long and short SSODNs and gene-linked templates both underwent gene insertion using the SSTR pathway, so they're much more sensitive to knockdown of FANK and HELQ than BRCA2 and RAD51. Right, so we really don't have an answer still on how we could improve insertion using single-stranded DNA templates. And so we sort of shifted gears, or in parallel, people were thinking about modulating DNA repair to improve this gene insertion. And so here the idea is that NHEJ and MMEJ are other competing pathways that limit your HR-based gene insertion. And so we were trying to see if you could find ways to limit NHEJ and so force the cell to go through HR. This is not a new concept, but for the past 5 years, some incredibly effective small molecules have come into the scene that are really efficient in blocking DNA PKCS or 53BP1 and improve gene insertion, sometimes by a factor of 3 to 4 in the left. And then blocking 53BP1 is a little less efficient, but you still get about a 20% to 30% improvement. And then the other concept is to block MMEJ, but this only works when you already block DNA PKCS. So you have to inhibit DNA PKCS and Pol theta at the same time to benefit from this strategy. And so we were looking at using these approaches to improve cDNA insertion. And so in the first study that came out last year, we were looking at just inhibiting DNA PKCS or inhibiting PKCS and Pol theta. And you can see that inhibiting both proteins did improve gene insertion quite a bit. However, when we inhibit both DNA PKCS and Pol theta, the toxicity is quite high. And so when you then account for improvements in yield, you benefit and you get a twofold improvement in yield when you block just DNA and PKCS. But if you block both of them, then you don't see any sort of improvement in yield. So that was not that useful. And so we kept going and we were looking at other synergistic pathways to inhibit non-HR pathways. And so this is work by Ma Su, and she is going to be presenting a rapid fire poster tomorrow, and I would encourage you to go look at her actual poster as well for more details. But in Su's project, Su was looking at combined inhibition of multiple different DNA repair proteins. And the combination that seemed to work effectively for her was the inhibition of DNA PKCS and 53BP1 simultaneously. So compared to the inhibition of either protein alone, there's a synergistic increase in gene insertion when you inhibit DNA PKCS and 53BP1. So we are able to get about 40% modified cells that express the complete CFTR cDNA when you block both of these proteins together. And unlike inhibition of DNA PKCS and Pol theta, you don't see any sort of significant toxicity with this sort of an approach. So this is in a range where now you don't need to think about enrichment, and that extra cargo space becomes available for some other more interesting cargo, which we still haven't figured out what that's going to be, right? And so then for the, you would be curious to see if this strategy allows you to replace or to restore the CFTR function. And so here on the left is an Using trace where you can see that the cells corrected using AZD, and UBV is just a different name for the HEP peptide. And you can see that these cells show restored responses to forskolin and CFTR inhibitor compared to the cells that were corrected without the use of these compounds. And on the right is a plot showing percent correction and CFTR function. And you can see that on average, corrected cells have CFTR function that's somewhat comparable to what you would see with wild type controls. And so, of course, I wanted to just end the talk with a little snippet of some ongoing work. So this is promising. However, one concern with the inhibition of DNA repair proteins is that you might make unwanted changes. And specifically with the inhibition of DNA PKCS, there was a Nature Biotech paper earlier this year which reported the presence or an increase in large indels at the on-target site when you block DNA PKCS, and that is a concern. The one point that we have sort of landed on so far is that the published work use single-stranded DNA templates, which are much less efficient compared to AAV templates, as you have seen in my talk. And so when we switch over to an AAV-based template, then what we observe is that there's a significant reduction in these large indels compared to the use of single-stranded DNA templates. So we see restoration of CFTR function, and we're using AAV templates, so we're hoping that we're not dramatically increasing the presence of large indels using our technique, but that is still ongoing work to evaluate the presence of large indels and safety concerns. So to summarize, I would like to end by saying that inhibition of DNA PKCS and 53BP1 together enables enrichment-free replacement of the CFTR cDNA. And this correction allows us to replace or restore the function of the CFTR protein in airway stem cells. And then we are still working on evaluating the safety of this approach, but so far it seems promising. And with that, I'm open to any questions you might have, and that's my contact.
All right, I'll start off the questions. So that was really nice to start. So you're doing a lot of the genetic editing, the gene editing in cells that are going through mitosis, like in the bottom of a dish. What is going to be the hurdle when you're going into post-mitotic cells? If you've thought about that, if you were going to do like an inhaled therapy, and how gene editing is just different there. Do you think this is going to be limited to putting cells back into the airway?
Right. So right now, HR-based systems are probably going to be limited to putting cells back into the airway. It's the efficiency of HR-based gene insertion in vivo in non-dividing cells is really low. And so that is a limitation. So there's a lot of work looking at the use of prime editing or prime editing combined with transposases and recombinases to replace the CFTR gene. And the idea there is that they do not rely on the endogenous machinery in dividing cells. But there, the efficiency is still not at the point where you can apply that in vivo. So that is the hurdle there.
Yeah.
Very nice presentation. I was wondering why you decided on cDNA, because without introns, it's likely to be silenced on the long term by methylations and so on.
Yeah, no, that's a great question. I think we did consider adding introns, but the introns were really large. And the second concern is that when you add an intron, the homologous repair machinery starts to identify the intron as a homology arm and tends to stop insertion there. So if you then are going to add an intron, it either has to be a random intron, like a HBB intron or something like that, that is super efficient but not CFTR specific, or you have to insert your gene in a different locus. So that's been done in the case of thalassemia. They put in HBB into HBA to rescue beta hemoglobin expression because they were going through this problem. Like they saw no protein expression if they didn't put in an intron, but they couldn't put in the intron back in the native locus, right? So there's some technical limitations. And in this case, since we see restoration of CFTR function, we're a little bit more hopeful that we can get away without using introns.
But you don't know how many passages, expression?
Yes. So, with this sort of, we've tested at least a couple of different passages. So we would passage our basal cells and differentiate them and look at function. And at that level, we don't see any sort of difference. Of course, ALIs only stay alive for a limited period of time. So within that limit, we don't see a degradation in that function.
Thank you.
Are there any other questions?
Great, thanks.
Thanks, Sriram, for a great talk. And we can stay up here because we're about to do a panel. All right, so we're going to have the speakers come up, and if there's any remaining questions that you have, you can ask the speakers as a kind of a panel. Ready? Come on up. Don't be bashful. And if there's no questions, then you can just stare at us under the interrogation lights that were behind you.
You can take your food. It's so bad.
Martin.
If I may, a question for Andre. I'm over here, Andre, in the middle. Never mind. Anyways, I was just curious with your ASO strategy, And sort of the efficiency do you expect out of the outcome? If you do this in your reporter systems, you have usually a relatively high level of mRNA present, but in native CFTR expressing cells, for example, it's a very low copy gene where we assume there's only, let's say, 2 to 5 mRNA copies in a cell or so. So how do you think this plays into the efficiency you can ultimately get out of your approach? Can you saturate with ASOs that basically every mRNA is bound to one of your read-through ASOs? Or?
Is this for me?
Yes.
I wasn't.
For Andre.
Okay, so I was trying to understand what you were saying because the reverb is. Could you mind if you step up in front of the? I didn't even know it was for me because the. Yeah.
Also, the light is terrible.
Yeah.
The question is for me, right? Okay, great.
It's really bright up here. Yeah.
This far.
When you're in your model system, you've tested it, you have usually like reporter genes or some relatively high expression level of your reporter. But if you go into the native CFTR context where you have only like 2 to 5 copies of mRNA probably in the subject to NMD, how do you think that is limiting your efforts that you would expect? And also, can you actually deliver enough ASO that you saturate that at least all the present mRNA is bound?
Yeah, that's a great question. So the question is whether, what do we know about the efficiency of RSO in the cell with the disease cells where the amount or the number of molecules of the CFTR mRNA may be low. And yes, we're constantly thinking about it. We just don't have the data yet. Our hope is that because we can affect the strength of binding, we did show, for example, in our system that if we use a double ASO, so we actually link 2 oligonucleotides, one binds downstream just to increase the binding efficiency, and the other one is the specific one, it actually does increase the efficiency of read-through in our system. So we're testing various approaches that would allow us to improve, the affinity of the oligo. So we hope, and we are in the nanomolar range in terms of affinities. So it's low 100 nanomolar, and we're just beginning. So we hope we can reduce that by affecting the sequence, the length, using various other structures. But yes, this is a concern that we are going to deal with when we're looking at cells. And we also can affect binding affinity by the modifications, which are known to, some modifications are further known to increase the binding. So we hope that eventually we will be able to target most of mRNA molecules, but I don't know the answer now.
We found in our studies that if you have very efficient read-through using suppressor tRNAs, that in fact, using SMUG1, which is an NMD inhibitor, has no benefit. So if you can get enough read-through, stabilize the transcript, and so you just need to get ahead of it, then the decay of the mRNA shouldn't be a factor.
Can you come forward?
I can do that. Or just,
Yeah, that would be the best.
If you can hear me.
Yeah, he's pretty loud. He's good.
Increasing the stability and the number of peptides made per CFTR mRNA in conjunction.
No.
You have?
I have.
Optimization.
Yes, and those results are, yes, and some of them work. And so so that is a viable strategy, at least in our hands. So when we're replacing the CFTR cDNA, we have been thinking about improving either the stability of the protein or how long the protein is open, things like that.
Well, I guess more in the sense that you have so built CFTR mRNA available, right? How do you stabilize that mRNA?
So we haven't been looking at that. We have been looking at the level of the protein itself.
Okay, well, maybe I can address that. So I just want to echo what John just said. So if you can induce sufficient read-through, because NMD is thought to depend on the binding of release factors, right, to the stop codon. So if you inhibit the binding of release factors, as many experiments with read-through compounds show, you do actually increase the stability of mRNA. So the hope is that there will be an increase in mRNA number of molecules if the read-through is efficient.
Yeah, and if read-through is efficient, so the current mechanism of NMD degradation, it's like positive feedback on the levels of messenger RNA, because if it's being read through, it does not undergo NMD through the pioneer cycle of translation.
Yeah.
If I recall correctly, when you were showing your data, I think it was for the Therapeutics ASO, and you were showing that, I think, for wild-type cells, you saw very minimal levels of effect on the splicing, so which would be reasonable since you only have the one mismatch. And I was just wondering if you looked at off-target effects of your ASO in mRNA in other genes, and if not, how do you propose to get into this?
Yeah, so we did look, and we didn't see off targets. We did it twice.
The question was if there's an ASO specific for the CFTR transcript, as shown in the talk, if there's a possibility that ASO will bind to other transcripts in the cell and have effects that are either toxic or otherwise. Yeah.
So first is when we design the antisense, okay? Then we have the option to choose the best one that will not have any, and we then analyzed the RNA. In splicing, the off-target effect is very, very minimal because it's not within the coding, okay? So it's, we don't see any off-targets. And maybe this can explain the safe toxicity profile that we saw so far, okay? Yeah. Yeah.
And to add to that, if you have an ASO that binds, in the middle of a coding RNA, that the ribosome is pretty good at pushing these off, and it shouldn't affect the expression of that protein. Yeah, it's like a train on a train track. It's a cow catcher of sorts. It'll just knock it right off. And so unless these ASOs are binding to maybe non-coding RNAs and affecting their function, it would be thought that they would have very limited off-target effects. Any other questions? It's really bright up here. If there aren't any other questions, oh my gosh, yeah.
So also in I guess it's the question of delivery, and we can debate delivery probably till the cows come home. But have any of you also thought about using, say, like the U7-based strategy, an expression of a small RNA to act on the ASO and to maybe get around some delivery concerns, kind of like the micro or the dystrophin skipping construct for children?
So the question is, you can use these U7 small RNAs, and this is a naturally occurring RNA that's really important for modifying the 3' end of histone transcripts, right? And they've made modifications where you can deliver them. They're short hairpins that bind proteins, they will then get moved into the nucleus, and you can put basically an Watson-Crick base pair ASO expressed with them in conjunction and affect splicing. So this is a approach that's being used for Duchenne muscular dystrophy, where many of the mutations cause duplications or loss of exons. And so has this been used for CF? I have not used it. It's a great idea. Then you'd be relying on likely viral delivery. And so it's just, you're putting your problems in another bucket.
Yeah, I guess that's kind of like the question I'm maybe getting towards is, do we think the delivery problems, is there, I guess, what are the pros and cons for delivery? That's the ASO strategy, say, are lipid nanoparticles and viral delivery, right? I'm trying to look ahead.
Yeah. So I think that that's a great question. I think one of the largest barriers to gene therapy or any therapy in the lung is delivery. The lung is really good at keeping things out. Otherwise, you have a pandemic, right? And so, yes, I think that the benefit with all of the approaches that have been talked about today is they actually use different delivery strategies. And so, it's, when you find one that works well, we might be able to use it for another approach. But yeah, till now, I think we haven't figured that out yet.
Well, there are studies showing that there is, somewhat successful delivery of antisense oligonucleotides used to, downregulate genes to the lungs. So, for example, my colleague John Watts recently published work showing that it is possible, and you see many studies here where people explore various formulations. So it may take time, but it's probably achievable. At least that's what I hope for.
Yeah. You have a question?
Mm-hmm.
Yeah. So the question is, can we take the ASOs and conjugate them to a carrier for better and more efficient delivery, not only to the airway cells, but also to other cells in the body for systemic delivery? So maybe getting at the pancreas or other organs that are affected in CF, or in conjugation with lipid nanoparticles, rather than just a naked nucleic acid approach.
Yes, so there are examples of conjugating oligonucleotides with, say, proteins, right, that bind to specific cell types, or with lipid-like tails that can drive delivery to certain tissues. So it is also a very, very active area of research. I mean, my lab personally is not there yet to explore these approaches, but it is definitely, something we're thinking about as well.
Maybe I will say that there is a balance between the exposure of other organs. Then you have to protect the ASO, package it, and hope that in vivo the ASO will be released efficiently. And the other side that we in splicing decided to take is to work with naked ASOs and try to target only the lungs. So if we will be, so far. So if we will be successful, and as I showed, we have very preliminary results to support this, maybe in the future we will be able to do so. But at the moment, it was important for us to see whether we can use antisense oligonucleotide naked in the lung and have some effect by our systems.
And there are efforts going on. There was one talk at Emily's Entourage, a group from Canada. They have developed an amphipathic peptides for the delivery of antisense oligos. And their data was compelling in a way that when they treated these cells, the uptake of ASOs was like, according to them, in seconds, which was remarkable. So there are efforts going on in terms of delivery of these antisense oligos, morpholinos, for W1282X and other variants.
Add that one of the biggest limitations to any of these delivery methods is endosomal escape. And so some of the most effective therapeutics that are now in clinical trials for myotonic dystrophy, Duchenne muscular dystrophy, it's estimated that less than 1% of what is delivered to a cell actually escapes the endosome. And that means that entity has to be really, really efficient once it gets into the cell. So there are groups that have developed endosomal escape vehicles, so it increases that barrier. And so these are like cyclic peptides and different charged molecules that will actually allow the nucleic acid to escape the endosome or rupture the endosome and get better delivery. And so it's really, I think, it's important to look outside of CF research and look into what other fields are doing, and then use that information to develop therapeutics for the lung and cystic fibrosis.
The other side that making escape faster also means you may have to deliver it more frequently, right?
Yeah, it gets into the half-life. Yeah. Yep. So you get a trickle of it from the endosome into the cell, and it allows for longer, basically sustained delivery. That's right. I think if you have something that escapes well and has a high half-life, you can get around that, or a long half-life. Any other questions? Not, well, thank you all for a great symposium, and hope you enjoy the rest of the conference. Thank you to the speakers.
Thank you. Your stuff is fantastic.